So first up is Jens Kremkow, a postdoc at Humboldt University in Berlin. Jens' talk will focus on single synaptic inputs, drive spiking in PV neurons in vivo. Jens will be followed by Richard Nod. Richard is currently an assistant professor at the University of Ottawa. His talk will focus on noise-gated dendrosomatic interactions, increases information transmission. And finally, we'll hear from Ben Scott. Ben is a postdoctoral fellow in the Department of Molecular Biology at Princeton University, and his talk will cover imaging global brain dynamics in transgenic rats with a miniature wide-field microscope. Please welcome Jens Kremkow.
All right, thank you very much for giving me the opportunity to present our update here, or my update. So what we've been working on for the last years is the question of why neurons in the layer 2-3 of somatosensory cortex or any cortices are so sparsely active. And this you can see here in this whole cell recording from a layer 2-3 cell pyramidal cell in a awake mouse. And you see that they spike like every other second. And this is quite remarkable because if you consider all the highly recurrent network that you have in cortex. So we were fascinated by this question, and an obvious candidate why it's so sparsely active are, of course, interneurons, and in particular, different types of interneurons. And the somatostatin interneurons have been suggested to play a role here because an activation of a single pyramidal cell can drive somatostatin cells. However, in general, you need a spike, like a trains-off spike, to drive these cells, in which, as you just saw on the previous slide, is not really what you see in these awake conditions. This is really what we were after, and our approach there was an in vivo approach. So we used two-photon targeted whole cell recordings and multiple neurons in cortical layer 2-3 in neurons. And if I say we, I have to stress that this was a team effort, and all the recordings were done by Jean-Sébastien, and I had the pleasure to look at the data. So he really recorded all the data, and this work was done in the lab of James Poulet. Anyway, so what Jean-Seb managed to do is he recorded with up to 4 pipettes in an anesthetized mouse, and neurons that he recorded were very close neighboring neurons. And with his method, he could, as I just said, record up to 4 cells simultaneously, and one of these traces is actually one neuron. So you see 4 neurons simultaneously, and due to the fact that it's in vivo, we had an active brain state. This was nice because now we could address our question by injecting currents and eliciting single action potentials in pyramidal cells, and then study the integration of monosynaptically connected and unconnected cells among these simultaneously recorded cells. And we did this for 3 different cell types. So we looked at monosynaptically excitatory input to pyramidal cells, to somatostatin, and to PV interneurons. And when we looked at, so these are examples that I show you now. When we looked at pyramidal cells, and this was in the active brain state, so not in the quiescent down state, but in the active brain state. And when we looked at an example of monosynaptic input to a pyramidal cell, we didn't see much. So you see here a few trials of the subthreshold responses. This is the raster plot, and this is the PSTH. And trust me, there's no mistake here. We love this figure. And this is the average EPSP. You see it was a monosynaptically connected cell pair. Similarly, for the somatostatin, there was not much. With a single EPSP, we couldn't elicit any somatostatin spiking. And most of our data that we have, the excitatory input was strongly facilitating. But strikingly, as the title already says, for the PV cells, it was completely different. So this is a PV cell that was driven by one single spike in the pyramidal cell, of course, multiple trials, and you see clearly that this cell, follows this synaptic input quite rapidly. And in fact, when we looked at the population, this was one example. When we looked at the population, you see that these responses are highly precise in time. So this is shown here, where this is the time after the presynaptic spike, and you see that this in the green is the high-resolution PSTHs, and you see these spikes happen after 1 or 2 milliseconds, and with a jitter of a millisecond, and they arise at the rising phase of the EPSP. So the black curve here is the EPSP. That was really striking. We didn't expect that in vivo. And also tells us something, like in other words, the PV cell is just copying the excitatory spike from the pyramidal cell. And if that is the case, we should see a time-locked di-synaptic inhibition in the cortical circuitry. And this is exactly what we found. So when we looked at, for example, unconnected pairs, so here I show a recording where we recorded 2 pyramidal cells, and we elicited spike here, and we saw a clear hyperpolarization following this pyramidal spike. And we see that also on the population level. And we see that for unconnected pairs, but also for connected pairs. So if we combine all our data together, so if we mush all our data together, we actually see a reduction in the spiking rate following an excitatory spike. So summing up, we think that this PV spike, single EPSP-driven PV firing can support this sparse firing. So this was the scientific update that I wanted to give. And then furthermore, I left the lab of James Pollet. So I started my own lab also in Berlin, actually in the same building, in the Charité and the Bernstein Center for Computational Neuroscience, looking at thalamocortical integration and functional connectivity. And I just want to mention it's because by chance the next European Neuroscience Conference is actually in Berlin next summer. In particular, in summer, Berlin is very nice. So you should take that opportunity, and I can show you around. Thank you.